So this lab 3.04, which you'll turn in here at 3.06, will take a couple of days to complete. The most important thing right at the beginning for you to do is to open up this student guide because it'll help you. It says do not turn this in. This is not what you turn in, but it'll help you work through it. And you start here. And one thing to keep in mind is that there's a section on the lab that has to do with using milk. That's actually, you can do that part of the lab, but the protein digestion, part C right here, is the one that's actually going to be what you'll turn in. And so if you come over here to the place where you turn in the lab a couple days from now, you'll see the link here to the lab report that you'll be turning in. And if you notice that, you're really only working with the protein part of the lab, part C, and you're going to put the pH levels that you're finding of the egg whites. And first you are going to take the pH samples, and then you're going to explain which one had the greatest amount of digestion in the order. This is the greatest, and this is the next. So digestion would be, you'd see that it fully the egg white, which is the protein, has completely digested or turned into liquid, basically, that it will then be used in our bodies. If it was in our stomachs, that would then become nutrients, and it would go down through our small intestines and continue to be absorbed into our bodies. So in the stomach is really where protein digestion takes place. And so that's what you're kind of exploring here, is what does it take to digest protein? So in other words, what kinds of things are in our stomach that actually digest protein? And so then you'll look at what's in each test tube and figure out what are those things that are really important to digest proteins in the stomach, and then which test tube is most like the real human stomach. So that's sort of the purpose of the lab. So let's go over here, and we'll go back to, it's a little tricky because this lab starts up here on day 1, and then you turn it in on day 2, and you actually come back 48 hours later to the lab. So we're going to you go through and you want to read all this. This is that student guide that we just were looking at, and talking about simulating the digestion in the stomach is what that page was just talking about. So we're going to go over to the virtual lab. You can view the tutorial. I'm going to start with begin the lab, and then this is a place where after you begin the lab and you do some of the work on it, you'll have to come back 48 hours later, and you're going to get a code to put in here so that you can come back in and look at the results. So what you'll need to do is you'll need to start, let's take a look at this. This is really what, I'm going to put this actually right next to that. And in fact, I might just stick this over here on the side here so that we have 2 windows open at once. Make it a little bit easier. Okay, so we're going to start. And it says that we're not going to do the milk one right now. We're going to start with the egg, and I'm hoping it'll let me do that. Come over here and you break up the egg. If you need to go down too, you go down like that. You pick up the egg with a spoon. There we go. And it says over here that I can't go over to the student guide, but you're gonna put 1 spoonful of egg in each of these beakers. So we got 1, 2. If you ever see that red with the you have to go until the red circle with the line through it is, you can't do anything if you're in that mode. So you have to find the sweet spot, I guess you would say, to get that in there. Okay, so let me put that down. Come over here. Using this spoon, put the egg in each of the 6 lab let's see, let me move this over a little bit more. The 6 test tubes in the rack on the right side of the lab bench. And then we need to fill it in, each test tube, 1, 2, 3, 4, 5, 6, with these substances. Okay, so let's do that. We will get, we need water. So let's get this pipette here. Come up and get some water. We need 10 milliliters of water. And we'll just go ahead and put it in here until we get 10 milliliters. Okay, that looks like 10. And we bring that over and we put it in, yep, where did that go? There we go. Pipette number 1. Put that in there. Now we need 10 milliliters of pepsin. So let's get pepsin. Get 10 milliliters of that. Yeah, it's tricky. And there we go. And we got to find that sweet spot again. Let's see here. There we go. Okay. Put that back down, find the sweet spot. There it is. Okay. Next we need 10 milliliters of hydrochloric acid. Oh, okay. Did I just do hydrochloric acid? Let me look here. I may have done that wrong, but when you go to do it, make sure you do pepsin first, and then hydrochloric acid is number 3. So in case you saw me do that wrong, can't remember. I'd have to go back, rewind this, but I just want to do a quick video so we can get you through the lab here. Okay, so then we bring that over. And that one. Bring it back. Now we want 10 milliliters of sodium bicarbonate. And yeah, this is too tricky. I should make this a little easier, but that's what it is. Okay, so bring that over. Number 4. Okay, and number 5. 5 milliliters of pepsin and 5 milliliters of sodium. Okay, so pepsin. Sodium bicarbonate, which is baking soda basically, which is very alkaline. It's not acidic at all. And pepsin would be an enzyme. Okay, so we got 5. Okay, bring that back, and we've got pepsin and hydrochloric acid that we're going to add. So this is going to be kind of the opposite of number 5. Number 5 was pepsin and sodium bicarbonate, so very alkaline, or base on the pH scale, high on the pH scale. This one would be hydrochloric acid and pepsin. So that's going to be super acidic with the enzymes as well that break down, pepsin breaks down protein. Okay, so we're going to bring that over. Let me move this. And last but not least, get the sweet, there it is. Yes, okay. Phew. Now, we need to use the forceps to pick up the pH strips. So we're going to take these forceps, and again, if you have trouble, you can always go down if you can't find the forceps. You come over here, and we're going to put it in the first one, and then we're going to go see what does it equal. So that one, you see the red circle will go off, and you'll put it in number 8. So you're going to put in for the pH test strip over here. Oh, that's the milk digestion, sorry. Let's see here. You're going to, this will be on your actual lab here. You're going to put pH of 8 for test tube 1. Okay. So let's get another pH strip. Put it in there. And that will be a pH of 6. Looks like it's landing on, even though it, so you can put that on number 2, even though it doesn't look like 6 because it's green, but let's put number 3. You got, that's going to land on a 1 here, so you can put 1 for number 3. Number 4, we've got, where will it let us land? Oh, number 12. So number 4, you put 12. Number 5, it's going to be, looks like it's landing on 5. So we'll put 5 for number, that's the 5th one. And number 6, let's go ahead and do number 6. And that's going to give us a pH of it's not letting us land anywhere, but that looks like a, oh, there we go. That looks like a 0. Yeah, it's a 0. So it's pretty low pH. So you're going to write that down in there. Let's go back to the procedures, and let's take a look at, it says place the stopper on all test tubes. Let's find that stopper. You have to make sure you get on that triangle there. And, oh, here we go. This can be tricky sometimes. I guess it's when the hand is more open is when you catch it. There we go. Okay, and then you put the stopper on, and then you're gonna, what does it say here? Once they've been covered, click Start on the timer, write down the code you've been given exactly, and copy and paste it into a text file that you can easily retrieve. For the reactions to take place, you're going to wait an entire 48 hours. After the 48 hours have passed, enter the code in the space provided, the one I showed you at the opening screen, and it'll bring you back to your lab where the reactions will have taken place. And that's when you're going to observe and record your observations. And we're going to just do it for the protein for the lab. I'm kind of giving you a shortcut for this lab because it's a lot of work. So what I always recommend is looking and seeing what the actual lab is that you're going to have to actually, the graded assignment you're turning in. It's a shortcut, but it's good to know, good way to navigate through this is you're noticing on the lab that you're really not having to answer the questions about the milk, so just do the egg white part. And then this is the part you're going to answer after you go through and do the timer. So let's just take a look and see about that timer. We're going to come up here, start the timer. Here it is. Here's our code. Whoa, it's crazy. So I would just copy and paste that somewhere, like maybe email it to yourself or put it in a Word document or something, and then later on you'll be coming back to the screen in 48 hours and you're going to be looking at these and putting in those results. So hopefully that helps. Let me know if you have any questions, and I'm always there at the Learning Lab when you need me. Just look at the schedule for whatever semester we're in, and I can always help in person as well.